So I'll share the screen. Hello, everybody. This is week 4 practice session for this course Cell Biology. And let me just share my screen so that we can actually begin with the lecture. So the template or the layout for this session is also going to be similar to what we have been doing for the past 3 weeks. Let me briefly introduce myself to the new learners that might be joining us. So I am Arveen Kaur. I am a PhD scholar at the Department of Biological Sciences and Bioengineering, IIT Kanpur. And along with this, I am also PMRF, and I am one of the teaching assistants for this course, that is Cell Biology, Cellular Organization, Division and Processes, that is being conducted by Professor Shikha Laloria from IISc, right? So in this concepts that will assist you in solving the week four's assessment that is still open, right? So I am not directly allowed to address that assessment because that will give you out answers, right? So, in spite of that, what we are going to do, we are going to go through the previous year's assignment, and probably there will be some similar concepts or some similar questions that might have been asked last year. And if we understand what is the rationale behind answering those questions, or what is the key concept behind those questions, we will be able to solve the this year's assessment as well, right? So that is going to be the first part. In the second part, instead of questions, what we are going to deal with is some of the key concepts, right? Some extra concepts, because NPTEL has very, very concise courses. So there's a lot of information being delivered to you, right? So it is not always possible while structuring these courses that uh, in-depth information about everything can be handed out within that concise video that is being recorded. So some of the concepts I feel are yet important for you to understand molecular and cellular biology also. And if like you are going to go and specialize in cellular biology or molecular biology, it might come in handy. So those are some concepts that we will deal with in the second half of this session. So yeah, we'll begin with the session. The first question that we are going to address from the previous year's assignment is, which of the following kinases is not required for mitotic events to take place? So the options are polo-like kinases, Aurora A, Aurora B, or none of the above. So you can think about it for a while. And since there are very little participants, I don't expect much, involvement with this. But you are most welcome to answer once I am done with saying out the question. If not, we will still discuss the answer. So the answer for this particular question is none of the above. Why? Because all of these kinases, Aurora family kinases, that is Aurora A and Aurora B, and polo-like kinases are both required and both important for mitotic events to take place. Like, right? Apart from this, there is also one major other family of kinases that is required for the mitotic events to take place, and that is which family? It is the MCDK family. So MCDK is also a kinase that is one of the most required ones for the initiation of mitotic events. Apart from that, these two family of kinases, polo-like kinases and aurora kinases, which have aurora A and B, are also essential, right? So we have gone through its details during ma'am's lectures. So I have taken one a snapshot from the same lecture by Professor Shikha Laloria, wherein the title was Other Kinases Important for Mitotic Events. Why? Because we have talked about the main kinase that is important for mitotic event is MCDK, right? So other kinases include polo-like kinase, which is important for assembly of bipolar mitotic spindle. It phosphorylates proteins required for separation of the spindle poles and early in mitosis, right? Then aurora kinase, aurora A and aurora kinase aurora B are also required. Aurora A is important in assembly and stability of the spindle, while aurora B is important in attachment of the sister chromatids to the spindle. Right? So this will also keep a check on whether these things have happened or not. These events have taken place or not. Um, yeah. Coming on to question number B. The key event that occurs at metaphase to anaphase transition is which one of these? Sister chromatids dissociation and segregation, sister chromatids association on the equatorial plane, sister chromatid condensation, or all of the above. So you can think about it. So for this, we have already dealt with metaphase also where while we were dealing with cell cycle phases, and we have also dealt with this metaphase to anaphase transition and the APC complex that is involved in it and how it works. That is what we did in, I think, week 2, right? So I hope you remember that. If not, I have dealt with these things in detail in week 1 and 2 practice sessions. You can go and watch the recording. For this particular question, the answer is sister chromatid dissociation and segregation. Right. So this is the key event that will drive the transition from metaphase to anaphase. Right. What were the other options? One was sister chromatid condensation. So when does the condensation for sister chromatid take place? It basically takes place in prophase or at least by the end of prophase, right? So pro or prometaphase is when sister chromatids will condense properly, right? After that, the next question is, and the next option was sister chromatid association on the equatorial plate. So yes, this happens in metaphase, but this is the key event of metaphase, right? It will not determine the transition from metaphase to anaphase, right? Only when all the spindles have correctly attached to the kinetochores of all these sister chromatids and it is ready for dissociation and segregation. That is when metaphase to anaphase transition will occur, right? So option A over here is the correct option. Okay. So that is what I have shown over here also, that in metaphase what happens is if we are talking about a cell, this is the equatorial plane wherein all the chromosomes that might be available in that particular cell have arranged in very like along the phase plate they have arranged, right? And this microtubules or spindles have come and attached to different sister chromatids, right? Then during transition, what happens? These spindles will basically shrink and they will pull these sister chromatids apart so that eventually in anaphase, they can be pulled apart and taken to different poles altogether, right? So that is what happens in metaphase to anaphase transition. Question number 3 is sister chromatid cohesion is established in which of the phases? G1 phase, S phase, metaphase, or anaphase? Now, this is something again that while I was dealing with this APC and how cohesion works, how cohesin and securin and separase work, Like when I had talked about this in that extra concept part of one of these lectures, I think week 2, if I am not wrong, we had discussed briefly upon this also. So I hope you have gone through the question. The answer for this one is S phase, right? So synthesis phase is the time when basically the sister chromatid will be formed in the first place, right? And if you remember, we have discussed this earlier that before the formation of sister chromatids, the cohesin particles they go and sit, or the ring they go and sit on the single DNA that has not yet been replicated, right? Once it replicates, automatically it has that ring around it, right? We will go through some illustrations that might also help tell this to us. Okay. So over here, this paper from PNAS, I'm not sure. I think it was a review paper, so you can go and look at it for better understanding. It was published in 2001 by Carson et al. And this basically talked about this establishment of cohesion, right? So they said that the establishment occurs in S phase or synthesis phase. After that, it is maintained until the metaphase of mitosis, after which it is dissolved, such that sister chromatids are pulled apart, right, during anaphase. I do have this illustration which I was talking about. It is not the same illustration that we had dealt with earlier, but let me just explain this one. Right. So what I said was that uh, much before S phase itself, before the DNA has replicated, right, before the sister chromatid has formed only, what happens is that these cohesion particles are loaded, or these cohesion proteins are loaded on that single DNA molecule that is present, right? Once this loading occurs, it is known as cohesion loading. It happens in late telophase or in the G1 phase, right? After this, what happens is during S phase, the sister chromatid is formed. And since this cohesion protein was already present, now these sister chromatids are linked to one another using cohesion, right? So this linkage happens in S phase itself. During prophase also, when the condensation for this sister chromatids is going on, they are linked together throughout the entire length, right? The entire arms of the sister chromatids are linked, right? Only after some time, after almost towards the end of prophase, when the condensation is complete and the chromosomes are moving to the equatorial plate of the cell, what happens is that the cohesion molecules from the arms along the length of the arms, they basically dissolve, and they leave these sister chromatids, but they are still strongly bound, especially in the center, wherever the centromere is. Like, it is not important that the centromere will always be in the center, it could be towards one end, but wherever the centromere is, over there, these cohesion molecules are still bound, right? And this binding breaks only when during anaphase, these sister chromatids are pulled apart, right? And how does that happen? Due to, because just before they are being pulled apart, there is this enzyme that is separase that is going to act on this, and this separase will basically cleave these rings, right? So once these ring proteins are cleaved, they can no longer bind these two sister chromatids together, and this force of spindle will also be pulling them apart, and once this cohesion protein is also broken, they will successfully be pulled apart to different opposite poles, right? So it is similar to what we have discussed before. Question number 4 is, which of the following class of proteins are not present in pericentriolar material? So I hope you remember what pericentriolar material is. It is abbreviated as PCM, if you remember that better. And the options for this question are cell cycle regulators, signaling molecules, microtubule organizers, or none of the above. So you can think about it a little. we talked about like in the lectures, pericentriolar material was mentioned um, when centrioles were being spoken about. Sorry. Yeah. So the answer for this particular question is going to be none of the above, right? So one thing that you definitely should know is because this the centriole is being talked about over here, and say pericentriolar material, also known as the pericentriolar matrix or PCM, is basically the scaffold proteins that are present around the centrosome and the centrioles, right? The microtubules are the major, majorly associated with centrioles, right? So they are always going to be there in the pericentriolar matrix as well, right? Apart from this, ma'am in her lecture has said that a lot of these are involved in a lot of signaling processes, and there are molecular motors that are carrying proteins from one place to another. That is also how signaling is occurring, right? So, cell cycle regulators and signaling molecules are also indeed present in case of present in the pericentriolar matrix, right? Coming to question number 5. Microtubules that overlap with each other when in anti-parallel orientation are called A. Astral microtubules, B. Kinetochore microtubules, C. Interpolar microtubules, or D. Intrapolar microtubules. So I hope you can recall this lecture wherein microtubules were talked about. Three major kinds of microtubules were actually talked about. So if you remember which three they were, you will automatically eliminate out one option at least, right? Out of those, you will have to recall which one can overlap with one another. So for that, if you remember the properties of these microtubules, it will be easy for you. Just look at that question. I will just have water and then I can discuss the answer for it. Yeah. So I hope I am visible. Um, Nonetheless, I am going to just present the screen once again because my view has also disappeared. Yes. Okay. So the microtubules that overlap with each other when in antiparallel orientation are called interpolar microtubules, right? So what were the three kinds of microtubules that were discussed in the lecture? They were astral microtubules, kinetochore microtubules, and interpolar microtubules. Intrapolar is one that we have not discussed, and it is not like no such microtubules are present when we talk in this context, right? So these are the three microtubules that you had to pick from. Let us, if I put an illustration, yeah, I have. So, what are astral microtubules? Astral microtubules are those which are basically originating from, all of these microtubules are originating from this microtubule organizing center, which are one of which is present on either pole, right? These have the centrosomes and the centriole and the pericentriolar matrix and everything, right? There are three kinds of microtubules that are originating. One is astral microtubules, which are originating and going towards the cell membrane or the cell wall, right? They are not coming towards the inner side of the cell, wherein the this chromosomes have actually aligned. They are radiating outwards, right? These are the astral microtubules. The other two are the kinetochore microtubules and the polar microtubules. So the kinetochore microtubules, as the name suggests, are microtubules that are coming inwards towards the chromosomes, and they are going and attaching to the kinetochores, right? These are the microtubules that are key for pulling these sister chromatids apart, right? During the metaphase to anaphase transition. Polar microtubules, on the other hand, are also radiating inwards towards the chromosome assembly, but they are not going and attaching specifically to the chromosome, to the centromere or kinetochore. Uh, and sometimes what happens is that these can overlap also, right? Big, and when they will overlap, they will run in an anti-parallel direction, right? Because in the microtubule organizing center, is the negative end, and towards where it is being radiated, whether it is outward in astral microtubules or towards kinetochore in kinetochore microtubules or inwards in polar microtubules, these ends are the positive ends, right? So these positive ends may overlap in case of polar microtubules, as is seen over here, and that basically answers our question. So this is what was asked over there. There is also this other schematic from nature reviews, wherein they are showing these different microtubules in different colors. So the astral microtubules are mentioned as AMT, right? As I said, all these microtubules have their negative ends to in the this the microtubule organizing center, wherein the centrioles and centrosome and this complex is present, right? And the positive end will be radiating outward, right? This is in green or this blue color, whatever it is the astral microtubule. Then in this bright red color are microtubules that are going and directly attaching to the kinetochores and the centrosome region, centromere region. These are the kinetochore microtubules, and then there are these microtubules over here, as you can see, are overlapping upon one another. These are the polar microtubules or the interpolar microtubules, right? Okay. So moving on to question number 6. The question is, which of the following proteins is absolutely required for metaphase to anaphase transition, right? So this is again something that we have discussed earlier. I hope you guys know the answer for this one. Option A is helicase, B separase, C glutaminase, or D phosphatase. Right. So which one of these from your memory can you recollect is absolutely required for metaphase to anaphase transition? Okay. So the answer for this is separase, right? We briefly discussed upon this while we were talking about cohesion molecules and the cohesion that is binding, and how after the activity of separase and contracting of the spindles, can only these sister chromatids be pulled apart, because separase will do what? It will basically break these cohesion rings, right? So, this is what I have picked from what we had discussed in one of the last sessions. That, for example, these white color structures or white color tubes or cylinders, they represent the DNA sister chromatids, right? So these are the sister chromatids, and they are basically bound by a ring or several such rings, which are basically cohesion molecules, or cohesins they are called, right? So there are multiple proteins that they are built up from, right? We will not go into the details of that, but what happens for if we want to separate these two uh, tubes or cylinders or sister chromatids, right? What will we have to do? We will first have to either remove this ring like this, or break this ring, or cut it, so that we can pull these sister chromatids apart. So for this, what is the enzyme that is required? The enzyme required is separase. But normally what happens? this separase is blocked by a phosphorylation and another enzyme, that is securin, right? So it is inhibited, it is in an inactive condition when it is interacting with securin. But what happens after the spindle have organized properly, the APC-CDC20 complex, they come and they phosphorylate the securin, and they ubiquitinate the securin so such that it is removed from the separase, and the separase will now be free, and it will kind of go and cut through or dissociate this cohesion ring. Right after it dissociates this cohesion ring, what will happen? These sister chromatids will not be bound together to each other anymore, right? So they will just be in their position, stationary. So if some more forces applied, they can be pulled apart, right? So before this, there was a physical barrier for their movement to different poles, but now that barrier has been broken, and broken by the activity of which enzyme? Over here, it is basically separates, right? Then comes question number 7, wherein the what is asked is the spindle pole body present in budding yeast is equivalent to which one of these? Is it equivalent to centrosomes or lysosomes or Golgi body or ER membrane? So this, I think, is a fairly easy one, because not all of these organelles we have discussed in this particular week's lecture, right? So even if you did not know the answer, probably you would, just because of that. But the key point is to remember this, right? Even when you are studying other organelles, this is something that should remain in your head. It might help you with other questions when more organelles are involved, right? So the answer for this one is centrosomes. Indeed, the centrosomes in mammals or higher eukaryotes is the same, or it is homologous to spindle pole body, which is present in the budding yeast, right? So I have taken this representation from again one of the reviews in Cell Division, wherein they have represented a centrosome and a spindle pole body. Right. So in the centrosome, like we have talked about, there is a mother centriole and then there is a daughter centriole. So in all, there are two centrioles that are present, right? Then it has multiple different kinds of appendages. They are linked to one another, but they are like perpendicularly present, right? There are microtubules that are involved in the PCM matrix, right? And then the components of the pericentriolar material, PCM, centriolar linkages, and intercentriolar linkage is what is given over here already, right? When we talk about spindle pole body, right, this is what is present in case of budding yeast, right? Over here also there is an this polar body, then outer polar body and the central polar body, right? And then there are different microtubules that are attached to these polar bodies. Also, I'm so sorry. Just give me a moment. I'm sorry, I was getting a call. Uh, I hope I am visible. What we were talking about is that the spindle pole body that is present in case of the budding yeast is equivalent to the centrosomes that are present in higher order eukaryotes, even mammals, right? They have the similar kind of basic assembly also, and the same function that they basically organize the microtubules, and that is how they are involved in many functions, even inside and outside of the cell cycle division, right? Then comes question number 8, which says the Frapp experiment on microtubules yields which one of the following inferences? Option A is individual microtubules are dynamic and their subunits are constantly being replaced. B is individual microtubules are constantly polymerizing at at the both ends. C is tubulin remains at the equatorial plate of the cell cycle. Or D, microtubules are static and stable in nature. So which one out of these was actually depicted or understood by the Frapp experiment that was done on microtubules? I will just share my screen again. Mhm. I hope you're getting the question. Okay, so let us just revisit uh, or visit the answer for this question. So the first one is actually the answer for this question, which is individual microtubules are dynamic and the subunits are constantly being replaced, right? So this is the correct answer. When we will discuss why this particular one is the correct answer. When we talk about FRAP, FRAP is a technique that it is a short form for this technique, that is fluorescent recovery after photobleaching. So, what does the name suggest over here, right? Just a moment. So, what does the name of the state technique suggest? That there are basically fluorescent particles or fluorescently tagged particles which are being given light or intense kind of signal, due to which the bleaching of this fluorescence particle is occurring, right? So once that is occurring, after that we are still kind of observing these particles at a particular space, right? After this bleaching has occurred, we have not stopped looking at this frame of reference, and we are still continuing to monitor it. And we see that after some time, fluorescence is coming back, right? So there is recovery after photo bleaching had been done. So first, let us go through the statement, then I will make some comments of my own. So, fluorescent recovery after photobleaching or FRAP is a fluorescence microscopy technique used to quantify two-dimensional lateral diffusion within a cell, right? So, in order to make you understand this particular statement, let us talk about how this fluorescence and photobleaching can work, right? So, what happens in case of fluorescence? What happens is that we fluorescently kind of tag some particles, right? There is one particle, uh, there is one kind of proteins present. Like there are multiple kinds of proteins that are present in a particular cell, but maybe we want to look at one particular kind of protein. So we will go and we will tag it with there are different kinds through which it can be tagged. For example, a simple, for reference, what we say is we can go and we can put an antibody against that molecule which goes and sits on this particular particle. And now only these proteins will have those antibodies that are tagged on it. And now we will go and we will put a signaling molecule which has a fluorescent intensity, which can go and recognize only these antibodies, right? So wherever these antibodies are sitting, therein this particle will go and it will also fluoresce. Right. So from that, we will basically be able to fluoresce the particular protein that we wanted to see. Right. So that is one way of how fluorescent tagging can be done. Now, what happens whenever this fluorescent molecule is being seen or observed, right? How will we observe these fluorescent particles? Obviously, we will observe it under microscope or under some kind of lens or camera, both of which will be involved, right? So what will happen whenever the this fluorescent molecule is excited, it will basically give an emission due to which we will be able to see it, right? Whenever it is going through this excitation emission cycle, it will lose its intensity, right? Because it is a fluorescent molecule, it will lose its intensity. And eventually we might not be able to see it anymore, right? So what happens in this case is that we will lose this fluorescence. But when we are looking at live cells, which have some proteins that are fluorescently tagged, right? So these cells will continually be moving also, and they will be performing their normal functions also, just that we can see some of those functions because we can see some of the proteins that are tagged with this fluorescent particle. We can observe them, okay, with a lens, a camera, etc. Right. So that is how we will perform recovery after photopleaching. Right. So, for example, there was one particular cell petri dish that we have taken. We have fluorescently tagged some protein particles and then we have photopleached it. So we have kept it under such strong intensity light that it got photo bleached. Now we are not changing our frame of reference. We are looking at the same area, right? And we are continuing to look at it for a while. If, for example, whatever of this photo bleached particle, it will not stay at the same place, right? We have not fixed the cells to the dish. They are living cells, so they are also going to move and perform normal functions, right? So if we are looking at that particular frame of reference only, we will see that, for example, if microtubules were stained, we will see that, okay, now after some time, like I am not aware of what this time period could be. Say about 30 seconds later, I start seeing some fluorescent molecules over there, right? And after maybe 5 minutes, if I look at the same frame of reference, what I am seeing is that all of these, the entire microtubule is again visible, right? How is this possible? We had given high intensity light and every particle had been photo bleached, right? And it cannot recover back. The same particle that has been photo bleached can now not fluorescence, fluoresce. Sorry for the tongue slip. So it cannot really fluoresce again. But instead, what can happen is that some other particle which still has that fluorescent property, it can come and it can replace this particle, right? So from this, we can basically, when FRAP experiment was done, we could say it that individual microtubules are dynamic. And how are they dynamic? Dynamic means they are constantly changing, right? They are not stable or static. They are constantly changing. And their subunits are constantly being replaced, right? That is how, after this recovery period was done, the microtubule again was visible because the particles that are now forming this microtubule are different from the particles that were earlier forming that microtubule, right? So that is why this statement also says that fluorescence microscopy, it is a fluorescence microscopy technique used to quantify two-dimensional lateral diffusion, right? The particles are laterally diffusing within a cell, right? Let us look at a representation of the same. So this was actually from ma'am's slide. I have taken this snapshot again, wherein they talk about the half-life of microtubules in mitosis. Uh, then we have talked about FRAP, wherein a laser beam is used to bleach microtubules labeled with fluorescent tubulin, and the recovery of fluorescence in the bleached region is monitored as a function of time. And as this graph can suggest, uh, the recovery is faster during the metaphase as compared to the interface, right? Over here, the microtubules might be more dynamic in nature than when they are in interface, right? So that is why the recovery is faster in metaphase. Also, I have kept another representation from one of the papers in microtubules, wherein there was a control that was taken, wherein if the microtubules are bleached, right, they are photo bleached. After bleaching, if we are observing this particular area, we will not be able to see this particular area. But after recovery, if we are saying T1 is maybe after 1 minute and T2 is maybe after 2 minutes, we see that in T1 some intensity has come back, and in T2 it looks normal now, right? But if there was a tubulin depolymerizing agent that was given in this particular setup, right? So originally, we would have some microtubules. After that, a depolymerization agent is given, right? And a photo bleaching is done. Once this photo bleaching is done, if the tubulin is like always depolymerizing, it will not be able to form a new tubulin, right? So the photo recovery, even if we will do, we will see there is a very faint or slight, this recovery of fluorescent molecules that we can see, and that is what is represented in this table also. That if in case of control, there was a normal, this can be considered as a normal photo recovery after uh, normal recovery after photo bleaching has been done, right? If, for example, a tubulin depolymerizing agent is given, then obviously this will be very slow, right? Or it and the percentage of fluorescence will go really down. But if tubulizing a tubulin polymerizing agent is given on the other hand, then this recovery will be very fast, because now they have something that will quickly catalyze this recovery of microtubules that are already present. So that is how the percentage of recovery will really drastically increase, right? So that is what we spoke about also. Question number 9 is select the correct statement regarding spindle attachment to the chromosomes. The first statement is sister kinetochores are assembled in back-to-back orientation to reduce chance of mono orientation. The second is cohesion at the centromere makes the paired sister chromatids to be more fluid in nature, or biorientation of the chromosomes is important to generate tension at the kinetochore between the sister chromatids. So which one of these is the correct options? Statement 1 and 2 are correct. Statement 2 and 3 are correct. Statement 1 and 3 are correct, or all of the above. This could be a slightly tricky one. So for that, you should have gone through the lectures very meticulously. You should have remembered some of the key points, which we will discuss when we are discussing the answer for this particular question. Uh, yeah, go through the options once, and we can discuss that then. Okay. So the correct option for this one is 1 and 3, right? So, which means that the statement B is not true. That is, cohesion at centromere makes the paired sister chromatids to be more fluid in nature, right? Obviously, this is an incorrect statement, but because what will cohesion do? At centromere, they will hold them together, or they will bind them together. They will not provide more fluidity or more dynamic nature to these sister chromatids, right? The other two, however, are correct. The sister kinetochores are assembled in back-to-back orientation. That is, if one is facing this side, the other will face that side, right? Why is this done? To reduce the chance of mono orientation, right? So that if both are facing this side, maybe the kinetochore complex also faces the same side, and the same side spindles come and attach to both, and both of them are pulled to the same side of the pole. That is not what we want, right? Then option number statement 3 was bioorientation of chromosomes is important to generate tension at the kinetochore between the sister chromatids. This basically means that they should be attached to spindles at two opposite ends, right? Then only this tension will be generated to pull them apart. If they are attached at the same side of the pole, it will not be so, right? So this was one of the snapshots from ma'am's lecture wherein bioorientation is being talked about. Let us go through these statements once. The first one is cystic chromatids in a pair must attach to opposite poles of spindle to ensure movements to opposite poles for equal segregation. To facilitate this, sister kinetochores are made in a back-to-back orientation that reduce the chance that they would both face the same pole. That is what we discussed, right? Then cohesion at the centromere also makes the paired sister chromatids in the CEN and pericentric region more rigid to maintain this opposite orientation of kinetochores, right? And then bioorientation is important for generating tension at the kinetochore that increases microtubule attachment to kinetochore. Yeah. Also, this is just a representation for showing like how it should be correctly attached, and what are the kinds of attachment, and what is the correct kind of attachment, but what is also the incorrect form of attachment, right? So if the microtubules, which are represented in black over here, are not at all attached to the chromosomes and the kinetochores in the chromosomes, then what will happen? There will be no tension that is created over here. But once they are correctly attached, they should not just be attached, they should be correctly attached, right? Then this tension will be created and they can be pulled apart, right? Then the B part of this figure represents different kinds of attachment, right? Amphitelic attachment is the correct form of attachment, right? So in amphitelic, what is happening? There is an equal attachment on both the kinetochores of the sister chromatids, right? So they can be pulled apart to different poles. In syntelic attachment, however, you can see that they are both attached to two different microtubules, but which are originating from the same pole, right? This is not what we want, especially in case of mitosis. In meiosis, however, we talked about this. This is how it happens, right? So over there in meiosis 1, wherein there are two sets of homologous chromosomes that are present instead of uh, this thing, instead of amphitelic attachment to the sister chromatids over here, over there syntelic or monotelic attachment takes place, right? So that I think we spoke about in lecture 1 or 2, right? So this is one form of attachment which is incorrect when we talk about mitosis. The other one is monotelic attachment. So in syntelic, there were two different microtubules coming from the same pole and attaching to two different kinetochores. In monotelic, there will be attachment to only one kinetochore through one microtubule that is originating from the sorry, centrosome at one pole. The final one over here is merotelic attachment. Is merotelic attachment, wherein what is happening is that although there was an attachment like amphitelic, wherein two different kinetochores had attached to two different microtubules originating at different poles, but one of the kinetochore had another attachment, which is the third microtubule that is also arising from one of the same poles as the other sister chromatid. So this kind of attachment, which is also problematic, is known as merotelic attachment, right? Which brings us to our final question. which says extended extended binding of net1 to CDC14 can lead to which one of these? Enhanced activity of CDC14, delay in mitosis exit, early entry into cytokinesis, or none of the above. So this has also been talked about. So the correct answer for this is delay in mitosis exit, right? So net1 actually binds to CDC14 and it inactivates or inhibits its activity as a phosphatase, right? CDC14 is what? It is a phosphatase, and when net1 is bound to this phosphatase, it will inhibit it from working. So it will be inactive, right? So thus the first option is definitely incorrect. And since there will be, if these are bound, then the mitosis exit can't happen. So there will not be an early entry into cytokinesis also. Uh, so B is actually the correct option, right? So this is one of the representations that I took from this paper by Shu et al., wherein they have very briefly drawn a complex. So where does this binding occur? The binding of net1 and CDC14 actually occurs inside the nucleolus, right? So during G1 phase till the anaphase, this net1 is bound to CDC14, and this binding is inhibiting the phosphatase activity of CDC14, and this complex together is present inside the nucleolus, right? After anaphase, however, there is an activation of CDC14, right? So it dissociates from the net. We don't exactly understand what this activating proteins are. Over here, they said that it could be TEM or CDC15. There are some papers that say SPO is one of the proteins that could be involved in this. So there are multiple players that could play this part, right? And once this activation of CDC14 occurs, what happens is net1 is phosphorylated, right? It will no longer be able to bind to the CDC14, and the CDC14, through its phosphatase activity, will like dephosphorylate CDC28 and cause its inactivation, right? And this will lead to the stopping of the kinase, the CDKs also, wherein the kinase activities will be inhibited, and thus it will eventually basically lead to an exit from this mitotic phase, right? So if this dissociation is not done, if it has an extended binding, that is net1 and CDC is bound for an extended period of time, then there will be a delay in the exit of mitosis, because the first the CDKs that are involved in the mitosis will not be able to dissociate properly, right? Okay. This was another representation that I took from the master's dissertation of Boran Erdene, which I found online. Right over here, also, for example, this person has represented that CDC14 and net1 are bound together and present inside the nucleus, right? But if this particular protein, CDC1 is activated, it will move out of the nucleolus, while net1 still remains inside the nucleolus and it is phosphorylated when CDC is not bound to it. And the CDC14 will basically drive mitotic exit, and one of the ways it will drive mitotic exit is through going and dephosphorylating CDC15, right? And this will be involved in the MEN, or the mitosis exit network. So that was it for the first part of this presentation, that was dealing with the previous year's assignment for week 4. We have a short lecture that is prepared for extra concepts of week 4, right? Over here, what we'll be discussing mainly is some of the events associated with mitosis, uh, um, like that have not been dealt with in depth uh, during the this week's lectures, right? Most of the materials that I have picked up and compiled in this particular presentation, all of the material, in fact, has come from this book, that is Molecular Biology of the Cell, Alberts et al. Right? So we'll be discussing that. Please allow me to have a very short break before that. So as I was saying, we will begin with the second unit or second part of this lecture practice session, which is dealing with some extra concepts that are majorly taken from the book Alberts, right? And they are kind of around the topics that have been touched upon by Professor Shikha Laloria in her NPTEL lectures. So let us begin with this particular session. Yeah. So the first topic that I wanted to discuss was actually the condensation of DNA molecules into chromosomes. Right, so we have been talking about the cell cycle phases. We have been talking about mitosis. We have been talking about chromosomes, sister chromatids, etc. But we have not really dealt with how these chromosomes came into existence, right? If at all. Uh, Although I am not starting at the basic DNA genetic level, but we have discussed about DNA a little bit in the previous lecture. Over here, let us discuss about chromosomes a little, right? So, eukaryotic organisms like you and I have special ways of packaging DNA into chromosomes, right? So, this is something that we are aware of. But let us go and look at how like efficiently this packaging is done, right? So if you have not thought about this before, or this thought has not entered your mind, let us give you an example. The example that I have directly picked up and quoted from this book, Albert's, right? And it is a very fascinating one. So, over here, we say that, for example, human chromosome 22 has 48 million nucleotide pairs of DNA, right? So, this single chromosome 22 has 48 million nucleotide pairs of DNA, right? We know that it is the largest chromosome that is present, right? So, if it is laid out as one long perfect double helix, the molecule would extend for about 1.5 centimeter if extended end to end, right? So, if these nucleotide pairs are present as a DNA helix that we normally talked about, like right in the last lecture, we discussed about how DNA is formed, how it is arranged, right? How it is packed, etc. Over here, it we are sorry, we have talked about DNA, how it is formed, and what is its chemical composition, and some other things about it. Over here, we are going about and talk about how it is basically being packed, right? So, if we consider this particular chromosome to just be made of these nucleotide pairs of DNA, So like we talked about in that left-handed helix or right-handed helix type of structure, it would, if that is how it was present, it will occupy 1.5 centimeter, right? If we had to look at it, right? Even though it was very compact, it would still have been 1.5 centimeter long, right? And do you think 1.5 centimeter is something that we can look at with our naked eye also, right? We don't really need a microscope for that. But can we actually look at DNA or chromosomes through our naked eye? That is no, right? And are even cells that big that they can actually hold 1.5 centimeter long DNA? The answer is again no, right? Cells itself are in micron length, right? Which is 10 raised to power minus 6 of a meter. Over here, like they even don't touch millimeters or centimeters, right? Except for some exceptions that we have. But cells are in microns of, like in a micrometer unit, we talk about cells, right? And nucleus is inside the cell. It is not even as big as the cell. And these chromosomes are present inside that nucleus also, right? So this 1.5 centimeter would not have been able to come inside a cell or a nucleus, right? But in fact, this book says that, but in fact, chromosome 22 measures only about 2 micron in length during mitosis, right? So mitosis is the time, and metaphase especially is the time when the chromosomes are most compact or most condensed, right? During this time, this uh, DNA molecule or DNA structure that would ideally have measured up to 1.5 centimeter measures only 2 micron, right? How is this possible, right? This represents basically an end-to-end compaction ratio of over 7,000 fold, right? So that is a huge, huge compaction. So the question that we ask and we will address today is how can this happen, right? So I'm sure some of you are already aware about this answer, and nonetheless, we are going to discuss this principle like we have discussed all other basic principles. How does this happen? This is basically a remarkable feat of compression, and it is performed by proteins that successively coil and fold the DNA into higher levels of organization. So there are some proteins that are involved in basically folding of DNA and its compaction, and that is what we are going to talk about now, right? So what are these proteins? What are these DNA binding proteins that form eukaryotic chromosomes? Right? These proteins are broadly divided into two classes. One is histone or histone proteins, and the other is non-histone chromosomal proteins. Right? Each of these protein classes contribute the same mass to a chromosome as the DNA. So what does that mean? When we talk about a chromosome, we automatically think about DNA, right? So DNA is coming and it is being wrapped around in such a way that it is forming a chromosome, right? But this chromosome actually contains, along with DNA, some proteins as well, right? These proteins are the reason that the DNA is actually folding and coming into that compact structure. Right, and they are saying that both these classes of proteins, that is histones and non-histone chromosomal proteins, are contributing the same amount of mass to a chromosome as the DNA. So one part of, like if the chromosome had to be divided into three parts, right? Three equal parts of mass. By mass, one part of mass comes through DNA, one part of mass comes through histone proteins, and the third part of mass comes through non-histone proteins, right? The complex of both classes of proteins with nuclear DNA of eukaryotic cells is known as chromatin. Okay. Okay. When we talk about the first class of proteins, that is histones, that we are mostly just going to deal with this histone proteins, right? The non-histone proteins are like They are of varied origins and like varied kinds of proteins are present in that class, and we'll not be able to deal with all of them, right? So we are not going into details about non-histone proteins, but over here we will deal with histones, right? So histones are proteins that are responsible for the first and most basic level of chromosome packaging, the nucleosome. The nucleosome is a protein DNA complex which was first discovered in 1974. So around 50 years ago, right? Around 50 years ago, this protein DNA complex, that is the nucleosome, was discovered. When interphase nuclei are broken open very gently and their contents examined under the electron microscope, most of the chromatin appears to be in uniform in the form of a fiber with a diameter of about 30 nanometer. So I have kept an image of an electron micrograph of chromatin, right? We will look at that. Uh, but right now we can just go ahead and read this entire points, right? If this chromatin is subjected to treatments that cause it to unfold partially, it can be seen under the electron microscope as a series of beads on a string. So what will it look like? It will look like is a string that is running over here. Let me just draw it. Right. So in this first, what they meant was that something like this we can see, like a very, very elongated version, just in, uh, please forgive those spikes that are coming, right? That is some defect with me trying to draw with my mouse, right? So apart from these spikes that are present, right? This, if we just focus at this particular region, this is how a chromosome will normally look like. But if we are basically subjected to some treatments, as I said over here, what will happen is that it will basically unfold into a string kind of structure, right? And this string kind of structure we will see that it has some beads on it, right? Something like this we will see, right? All along the entire length, a similar structure will be seen. This is referred to as beads on string, right? So over here in the first representation, I have drawn something and then I have unfolded that, right? It is a string that is really wound tightly across one another. Once it is treated with some chemicals, this binding could be lost or unfolded, and then a very, very long string will appear and it will appear to have some beads on the string, right? The string is DNA and each bead is a nucleosome core particle, right? That consists of DNA wound along a histone core. So we are saying that these histones are basically going and binding around, is basically a core protein around which DNA is coming and binding, right? And this histone has a core protein, which is a core particle, which is known as nucleosome core particles, which is made up of different kinds of histones that are coming together and making an octameric unit. That is an 8 histone unit is it is making, right, around which DNA is coming and binding. So we will look at that also. So this is what I was trying to draw over there. So in the first case, if we just look at a chromosome, this is a chromatin from interphase, right? If we look at a chromatin in interphase, this is how it will look like. In figure A, right? It is an electron microscope image that is present, right? But if this chromosome was chemically treated, right, such that unwinding occurs, what will we see? We will see a string kind of structure which has these small dots on it, right? So the string, as we now know, are the DNA, and these dots or beads that are present around it is the nucleosomes, right? And these nucleosome core particles are present or consistent of 8 histone proteins that are bound together. So it is an octamer made of different kinds of histone proteins, right? So we can study a little about that also. We will. And over here, first, since we were talking about histone, there are some non-histone proteins also, like we said earlier. In this, you can see that these circles, the perfect circles with those four compartments in between, they represent the histones, while the non-histone proteins are present like along the histone core particles also, and on the free DNA strings also. So there are various kinds of these histone non-histone proteins that are there, and they are present on very different kinds of locations, right? So they have very varied functions also to perform. So this is an illustration wherein chromatin consisted of DNA bound to both histone and non-histone proteins is present. The mass of the histone protein present is about equal to the total mass of the non-histone proteins, right? So I hope you can make that out. That non-histone proteins, although they are different in nature, they are also just as abundant mass-wise as the histone proteins, right? In total, the chromosome is about one-third DNA and two-third protein by mass, right? Because we had said that we have divided the chromosome into three parts, which are equal in mass, right? One of them is DNA, one of them is histone proteins, and one of them is non-histone proteins. So altogether, when we look at it, chromosome is one-third DNA and two-thirds protein by mass, right? So over here, uh, let us briefly talk about how exactly histones were discovered, right? So what happened was that once there was some chemical treatment given to the molecule such that, or to the chromatin such that this string was discovered wherein beads were present, like interspaced at some distance, right? It was seen after some kind of chemical treatment was first given, right? After this, what was done was that scientists, they used a nuclease, right? What will a nuclease do? It will go and basically disconnect, or it will go and cleave a nucleic acid, right? So over here, there is one kind of or one part of the nucleic acid that is bound around this histone core particle, that is yellow in color, and the nucleic acid that is bound around it is red, right? But there is also some free string of nucleic acid that is present, right? So once nucleus digestion is done, what will happen? It will go and digest these strings that were free, right? Once these are cleaved and they are digested, what will happen is that the nucleosome core particles will be released, right? They will be detached from one another and they will also be released. After that, dissociation of these core particle can be done with high concentration of salt, right? With that, what will happen? We will basically obtain DNA separately and the octameric histone core particles separately, right? Once this was done by scientists like back in 1974, what did they see? That the DNA double helix more or less was always 147 nucleotide pair, right? So there is a definite amount of DNA that can actually wind around one particular histone, right? It might be variable from within different stages, but this is like the most general form that they could find, right? And when they actually dissociated this octameric histone core, what did they found? That this histone core was made of two tetramers, right? Now, these two tetramers, the first set of tetramer, which is represented over here, had H2A, two H2A molecules and two H2B molecules, right? And these together came into a complex and formed a tetramer, right? The other tetramer, which is depicted in green and blue over here, consisted again two H3 histone molecules and two H4 histone molecules, and these also came together in a complex and formed a tetramer. Then this complex tetramer and this complex tetramer came together and formed an octamer, which was a octameric histone core particle, right? It is very, very conserved, right? And I don't know if I have written it over here. I don't think so. But these histone proteins, again, they are proteins, right? So they are also made up of amino acids. And these histone proteins are actually very small protein particles when we talk about them separately, like not in a unit, but separately when we talk about it, they are very small protein particles, right? And what is one of the characteristics of these protein particles is that they are very, very and lysine rich, right? So these are the basic amino acids that are usually present in histone core complexes, right? So they are arginine and lysine rich. That is something you can which might help you in your competitive exams maybe later, right? Okay. So, speaking about nucleosomes, let us discuss a little about them. Nucleosomes repeat at intervals of about 200 nucleotide pairs, right? So, 147 is wrapped around the histone core particle. Then there is some that is free that can be up to like 40 base pairs. It is ranging from around 40 to 80 base pairs, I think. So, around at after every 200 nucleotide pairs, there would be a new nucleosome. So cell with 6.4 into 10 raised to power 9 nucleotide pairs contain approximately 30 million nucleosomes. So they are that abundant in our cells, right? The formation of nucleosomes converts DNA molecule into a chromatin thread about one third of its initial length, right? Yeah. Also, all four of the histones that make up the core of the nucleosome are relatively small proteins, like I told you, that they are only like 100 to 135 amino acids long, and they all share a structural motif which is known as a histone fold, which is basically how they came to be known as the histone family protein, histone protein family, right? Then the DNA helix makes 1.7 tight turns around the histone core octamer. So we know that around 147 nucleotide bases are present that around that nucleosome or histone core octamer. But how many turns does the DNA helix make? It makes around 1.7 turns, right? Okay. So this is another very, very interesting, phenomena that I thought I will discuss. So, okay. Just think about a nucleosome. So a nucleosome is kind of like a round shaped, more or less a spherical shaped protein that is present, right? So for DNA to be wrapped around it, there have to be some structural changes or some bends that have to occur in the DNA also, right? So DNA is not really formed in a way that it can be bent like any in any direction or in any way, right? But over here there have to be some bends that should be made so that it can go and it can actually wrap around a nucleosome. So how is this taken care of, right? So if you think about it, there are some pieces which like allow bending and some that are a little rigid, right? The rigid forms are G and C, right? Because they have triple hydrogen bond, they are more stable. So if we talk about G and C positive regions or G and C predominant regions, they will not be able to bend properly around the histone core particle, right? On the other hand, if we talk about nucleotide particles or nucleotide components such as AA or TT or TA dinucleotides, these have slightly less rigid kind of structures, right? They can bend a little, right? If they can bend a little, these are the kinds of dinucleotides that are preferred in the minor groove regions that can basically turn around and cause bends in the DNA so that it can wrap around the nucleosome, right? So over here, this yellow represents the histone core particle of the nucleosome. It is the histone octamer, like we talked about. The red structure, the double helix, is the DNA of the nucleosome. And you can see that it has to be bent a little, right? So it is usually bent around the minor grooves, and these minor grooves should actually be grooves which are rich in AA or TT or TA dinucleotides rather than GC dinucleotides or GG or CC nucleotides, because they have a stronger hydrogen bond, and they are a little more rigid than these dinucleotides, which are actually preferred for sites wherein bending has to occur, right? So the bending of DNA in a nucleosome is kind of like this. And this can also kind of make you think that this means that after about, like, it is not like a set number that okay, after every 200 nucleotides there is a nucleosome core particle, then after 200 there's again another nucleotide core particle. That is not the case, but roughly around this approximately 200 nucleotides. It could range from maybe 160 to maybe 210. I'm not sure about it, right? But it is a range, it is not a fixed number. So in this range, there will be a repeated expression of some sites which are rich in these kinds of nucleotides, which can actually cause bending on this nucleotide on this nucleosomes, right? And there will be some which will be very rigid and can't be bent over. So there will be kind of a similar identity that is spread across the entire DNA, right? So it is very different from person to person, yet it is somehow, in some senses, it is really conserved, right, to perform these very, very basic tasks. Okay, after having dealt with the heavy part for this lecture, we will come on the lighter version, which is a recap for mitosis, right? So mitosis is again something we had dealt with in maybe lecture 2 during the practice session 2, right? Let us just recap it a bit. So this is a fresh representation that I have again picked up from this book, Albert's, right? And in prophase, which is the first phase of mitosis, what happens? What does the cell look like? Right? So we can see that there is a nucleus that is still present. There is a intact nuclear envelope. Right? Then there is some condensation of the chromosomes. So there is condensing replicated chromosome consisting of two sister chromatids which are held together along their length. So over here, the cohesion molecules are also present along the entire length, right? Then kinetochores are also visible, and centrosomes are also visible, and they are going to start moving towards opposite poles, right? And mitotic spindle has also started forming from the centrosomes, right? So at prophase, the replicated chromosomes, each consisting of two closely associated sister chromatids, condense. Outside the nucleus, the mitotic spindle assembles between the two centrosomes, which have replicated and moved apart. For simplicity, only three chromosomes are shown. Uh, yeah. In diploid cells, there would be two copies of each chromosomes present. In the fluorescence micrograph, chromosomes are stained orange over here. Actually, you can better identify it as red, I think. And the microtubules are green, right? So these are the chromosomes. They are still present inside the nucleus, right? So they are really in that defined, boundary of a nucleus. They are still present, but they are condensed. We can see that. And the green is a microtubules. In the pro-metaphase, which is a late prophase or an early metaphase, the centrosome have moved towards the spindle pole, and the nuclear envelope has fragmented, and the kinetochore microtubules have also started joining to the some of the microtubules, right? And the chromosome is in active motion, right? So it is going here and there so that it can finally come and attach to and align at the metaphase plate. So prometaphase starts abruptly with the breakdown of nuclear envelope. Then chromosomes can now attach to the spindle microtubules via their kinetochores and undergo active movement, right? So over here, as you can see, the microtubules are still very well visible, and now the chromosomes are not restricted to the nucleus because a nuclear envelope has broken down, and they have basically moved apart, right? And they are now moving along to find that where do they have to align along the metaphase plate, right? Next comes the metaphase, wherein the centrosome wherein the centrosome are at the spindle pole and the kinetochore microtubules have kind of attached, right? At metaphase, the chromosomes are aligned at the equator of the spindle. Midway between the spindle poles, the kinetochore microtubules attach sister chromatids to opposite poles of the spindle, right? As you can see over here. Uh, Red over here is again representing chromosomes which are present at the equator or at the metaphase plate, which is midway between the two opposite poles, right? Just a moment. Yeah, so it is midway between these two opposite poles, and the microtubules are coming and attaching to the sister chromatids, and this overlap of red and green color you can see over here and appreciate it as this yellow that can be seen, right? So after this, what happens is during anaphase, the daughter chromosomes are basically pulled apart and they are pulled towards different poles, right? The spindle pole is also moving outward and there is shortening of kinetochore microtubule that is occurring, right? That is how these sister chromatids are actually being pulled towards these poles, right? At anaphase, the sister chromatids synchronously separate from two daughter chromosomes and each is pulled slowly towards the spindle pole it faces. The kinetochore microtubules get shorter and the spindle poles also move apart. Both processes contribute to chromosome segregation. So we know kinetochore microtubules were the microtubules that were directly going and attaching to this kinetochore complex, which was present on both the sister chromatids, right? During anaphase, what is happening is that these kinetochore microtubules are now shortening such that whatever they were attached to is being pulled apart, right? So they were attached to chromosomes over here. Once there is shortening, right? And simultaneously where they are attached to over here, the centrosomes, they are also moving apart, right? So there is shortening also and they are also moving apart. So the sister chromatids automatically will move apart to different poles, right? Then in telophase, you can see over here, there's a set of daughter chromosomes at one spindle pole and a similar set of daughter chromosomes on another spindle pole. The interpolar microtubules are the only ones that can be seen in the middle right now. The centrosomes are still present and the contractile ring has started to form. Right between these two different poles, there is a contractile ring that has started to form. Also, nucleoenvelope reassembly has started around individual chromosomes, right? So during telophase, two sets of daughter chromosomes arrive at the poles of the spindle and they decondense, right? A new nuclear envelope resembles around each set, completing the formation of two nuclei and marking the end of mitosis. The division of the cytoplasm begins with contraction of the contractile ring, right? So over here also you can see there's a faint this distinction that is present. These are the two opposite poles and some kind of from here you can see it has started becoming a little round again on both ends, right? Because the nuclear envelope is also slowly forming again. Once it is completely formed again, these chromosomes will be present in that inside the nucleus and they will appear in a more rounded morphology. And then comes cytokinesis, which is the final phase of mitosis, wherein the nuclear envelope has completely surrounded decondensing chromosomes, right? So chromosomes are also not as condensed as they were in the earlier phases of mitosis. Now they have also decondensed a little, and the nuclear envelope has formed around them. Also, contractile ring creating has created a cleavage furrow, and there was reformation of interphase array of microtubules nucleated by the centrosome. During cytokinesis, the cytoplasm is divided into two by a contractile ring of actin and myosin filaments, which pinches the cell in to create two daughters, each with one nucleus, right? That is what is happening over here. That is what can also be seen in this micrograph, right? And as we talked about, now the chromosomes are within the nucleus and they appear more like in a rounded kind of morphology. So when we talk about mitotic spindle, right, these are the microtubules that had been talked about for so long. The central event of mitosis, the chromosome segregation, depends in all eukaryotes on a complex and beautiful machine which is known as a mitotic spindle. So I think all of you will agree to this particular point, right? Because if the microtubules are not present, the metaphase plate alignment will not occur, then the metaphase to anaphase transition can't happen, right? Then the chromosomes will not be pulled apart, and this very sophisticated array of events cannot occur anymore, right? Then the spindle is a bipolar array of microtubules. Why are we calling it as a bipolar array of microtubules? Uh, just a moment. Am I audible? My screen froze. I will just share my screen again, okay? I think I might be audible only. Okay. Yeah, we were talking about these mitotic spindle being bipolar, right? So why are we calling it bipolar? Because we talked about them having these negative ends and positive ends. The negative ends were always towards the centrioles in the centrosome. The positive ends were always radiating outward, whether it is towards the poles or towards the chromosomes in the middle, right? So that is why we are saying that spindle is a bipolar array of microtubules which pulls sister chromatids apart in anaphase, thereby segregating the two sets of chromosomes to opposite ends in the cell, where they are packaged into daughter nuclei, right? The core of the mitotic spindle is a bipolar array of microtubules, the minus end of which are focused at the two spindle poles, and the plus end of which radiate outwards from the pole, which is what we have been discussing, right? Um, was there some other change? Ah, okay. Yeah. So the plus ends of some microtubules, which are known as interpolar microtubules, overlap with the plus end of microtubules from the other pole. These result in anti-parallel array in the spindle midzone, right? These are the only tubes that can microtubules that can run anti-parallelly, right? These are represented in red, that is interpolar microtubules, and as you can see, they have some overlapping segments in the middle, right? So these can only be seen in case of interpolar microtubules. The plus ends of other microtubules, that is kinetochore microtubules, are attached to the sister chromatid pairs at large protein structures called kinetochores, right? So these are the kinetochore microtubules. They are represented in blue, and you can see that they are attached to these large protein structures, kinetochores, which are represented in red over here, right? And these are located at the centromere of each sister chromatid, right? Then many spindles also contain astral microtubules, which are depicted in green over here, which radiate outward from the poles and contact the cortex, helping to position the spindle in the cell. right? So this, what is his astral microtubules doing? They are basically going and looking at where the walls of the cell are, and according to that, they will position this microtubule organizing center also, and the other microtubules that are radiating out will also come towards the middle of the cell, till at least the middle of the cell, right? That is how they will feel like what is the dimension of the cell, where or how it should be organized, etc. Right? This is the representation of the same. Then chromosome attachment to the spindle, mitotic spindle in animals, right? So when we talk about mitotic spindle in animals, during late prophase, like we talked about, the nuclear envelope is still kind of present. Only during very late prophase or early metaphase does this nuclear envelope start dissociating or fragmenting, right? Till then, the spindle, these centrioles, have started to move towards different ends. But at the same time, what is also happening, they are still on the outside, right? They have not been able to go anywhere near the chromosomes, which have already condensed inside, right? In early prometaphase, there is lateral kinetochores that start arising, right? Over here, now they can enter, and lateral kinetochore attachments form, the chromosomes arms are pushed outwards, right? Then during mid-pro metaphase, end-on attachment occurs, right? To some ends, the kinetochores start attaching. And once they start attaching, that is how these microtubules or mitotic spindle actually align these chromosomes at the equator, or during the metaphase, which is also known as biorientation, wherein two different sets of microtubules will attach to different kinetochores in the sister chromatid, and they will also be like attached on the other end to opposite poles, opposite centrioles on opposite poles, right? So this is how chromosome attachment to mitotic spindle in animals occurs. That brings us to the end of this particular segment also. So is actually all that I had prepared for this segment. That is just how far I could go for this particular meeting. Right. Yeah. So in that case, that brings me to the end of what I had prepared to be delivered in this particular lecture, right? If you guys have any problems or doubts that I can possibly address over here, I will be more than happy to. If that is there, then you can just turn on your mics, speak up. If there are multiple people who want to speak up, maybe you can raise your hands first. If not, if all your doubts are solved or you don't have any, we will end this meeting a little earlier only today. And then in that case, we will meet next week during the same time on Sunday, 3 to 5 p.m. With week 5 lectures, PYQs for week 5 lectures, and some extra concepts dealing with that week's content. So that is it from my end. I am assuming there are no questions then. Okay. In that case, thank you guys. I will end the recording and then we can all leave the meeting. Thank you for staying till this time. Thank you for attending the session.